Good evening to all the CUET aspirants. So, here we are in discussion with double membrane bound organelles. So far we have started discussion on mitochondria and we have landed up to electron transport system. Today we will look into the electron transport system of mitochondria, followed by D-loop replication which happens in mitochondrial DNA, followed by chloroplast DNA, and then comparative analysis between chloroplast DNA and mitochondrial DNA. Look into the session clearly. First, about electron transport system. Let us recall the class of yesterday's, wherein we have learnt that the glycolysis which happens, of course in biochemistry we will learn that in detail, but as of now you remember this part, that in cytoplasm, firstly in cytoplasm what takes place? Glycolysis occurs. Glycolysis occurs in cytoplasm, where glucose breaks down to two molecules of pyruvate. Glucose breaks down to two molecules of pyruvate, and this pyruvate will directly land into the mitochondrial matrix. Okay, pyruvate directly lands into mitochondrial matrix. Fine, right? Inside mitochondrial matrix, pyruvate will first undergo oxidative decarboxylation. Okay, pyruvate first undergoes oxidative decarboxylation, giving out 2 NADH for 1 glucose. For 1 glucose, 2 oxidation decarboxylations will happen and it will give out 2 NADH and acetyl CoA will be formed. And then inside mitochondrial matrix itself, TCA cycle or Krebs cycle will take place, wherein for one cycle it will be giving out 3 NADH, 1 FADH2 and 1 ATP, and then it also gives out 6 NADH, 2 FADH2 and 2 ATP. Clear, right? So, here mitochondrial matrix is rich with NADH and FADH2. Fine, right? Now, oxidation of NADH and FADH2 and giving out the cellular energy, that is ATP, that takes place in mitochondria. So, here if you observe firstly, in inner mitochondrial membrane, that is inside the cristae, what happens? All the complexes, electron carriers, protein channels, whichever are required for simplifying the complex energy to the simplest energy, that is ATP, are present inside the inner mitochondrial membrane. As such, there are 5 complexes to be learnt. What are those 5 complexes, ma? First, complex number 1. Yeah, first complex is NADH dehydrogenase. First complex, NADH dehydrogenase. Second complex is succinate dehydrogenase. Succinate dehydrogenase. And then third complex is NADH dehydrogenase, succinate dehydrogenase, and complex 3 is cytochrome BC complex. And complex 4, cytochrome C oxidase. Cytochrome C oxidase. And complex 5 is ATP synthase. ATP synthase or F0 F1 complex, F0 F1 complex is also known as oxysomes or Fernandez-Morgan particles. Keep noted, we will get questions with different synonyms. Okay, complex 5 which is ATP synthase, F0 F1 complex also known as Fernandez-Morgan particles or oxysomes. So, here if you see in mitochondrial matrix, wherein first NADH will be oxidized to NADH plus protons plus electrons, okay, by the complex NADH dehydrogenase. this complex 1 NADH dehydrogenase will phase from matrix, mitochondrial matrix to intermembrane space. This pump or channel will transpan both the sides of mitochondrial membrane. It will transpan both the sides of inner mitochondrial membrane, wherein in the mitochondrial matrix side NADH will oxidize to NAD plus. So here complex 1, that is NADH dehydrogenase, it will give out NAD plus protons plus electrons. Remember students, where electrons are accepted by first electron acceptor, that is flavin mononucleotide of complex 1, whereas protons are directly excited or pumped from mitochondrial matrix to intermembrane space. Fine, right? So here FMN accepting the electrons, it will give out to ferrous sulfur complex. Okay, ferrous sulfur complex will again pass out the electrons to mobile electron carrier, which will carry the electrons from complex 1 to complex 3, and that mobile electron carrier is ubiquinone. Okay, ubiquinone accepting the electrons will turn to hydroubiquinone or ubiquinol, you can say. And then if you see complex 2 is succinate dehydrogenase. Unlike complex 1, which is transpanning two sides of the mitochondrial membrane, complex 2 will not transpan two sides of the membrane. It will phase from the mitochondrial matrix to inner mitochondrial membrane only. It is not up to the intermembrane space. It is not transpanning both the sides. So what happens here is, flavin adenine dinucleotide is the molecule which will accept both the electrons and protons here, wherein from succinate to fumarate, which is one of the step from Krebs cycle, where FADH2 is oxidized. FADH2 is oxidized to FAD plus protons and electrons. Here both the protons and electrons are delivered to FAD only, flavin adenine dinucleotide, and to ferrous sulfur complex. One strong remark to be remembered is, for both complex 1, complex 2 and complex 3, ferrous sulfur complex is present. Okay. So here ferrous sulfur complex from complex 2 here, it is giving both electrons and protons to ubiquinone itself, and this also gives out both electrons and protons, and it turns to hydroubiquinol. From complex 3, that is cytochrome BC complex, protons are first sent out to the intermembrane space and electrons are given out to cytochrome B of complex 3. Remember that. From here electrons are again passed to ferrous sulfur complex, followed by cytochrome C. Cytochrome C is again a mobile electron carrier. See, remember students, there are two mobile electron carriers in the ETS of mitochondria. One mobile electron carrier is ubiquinol, that is from complex 1 to 3 and 2 to 3. And next is cytochrome C. This is the mobile electron carrier which is facing towards the intermembrane space. Okay, cytochrome C is facing towards the intermembrane space, and this gives out the electrons. This is being transported to complex 4, which is copper molecules, where copper molecules are present in complex 4. 1, 2 and 3 ferrous sulfur particles are present. In complex 4, copper molecules will be accepting the electrons, which will give out to cytochrome A3 of complex 4, which is cytochrome C oxidase complex. At the end, in the mitochondrial matrix, one molecule will be in a long wait for the acceptance of electrons, that is oxygen. Oxygen immediately accepts the electrons, binds with the protons which are present in the mitochondrial matrix and turns to water molecule. Always do remember, one electron will be moved by two protons. Okay, one electron will move along with two protons. As such, when two electrons will bind to oxygen molecule or atom, what happens is 4 protons will come here, of which 2 protons are required for water molecule formation. Rest 2 protons will be excited again to intermembrane space. Now what happens here is, there is an electrochemical gradient that is generated in mitochondrial matrix. After water molecule formation, there will be a deficit of protons. Okay, a proton gradient proton gradient is generated in mitochondrial matrix. Here what happens is, where protons will be deficient after the water molecular formation, whereas in intermembrane space it will be rich with protons. Okay, intermembrane space will be rich with protons and mitochondrial matrix will be deficient of protons. From intermembrane space, the protons will run to ATP synthase complex from intermembrane space to mitochondrial matrix, where F0 F1 complex is there, right? F0 will be embedded inside the inner mitochondrial membrane and F1 will be facing towards the mitochondrial matrix. So, here from intermembrane space to mitochondrial matrix, the protons are pumped. For the formation of 1 ATP, 4 protons are required. So, 4 protons are pumped in a run and 1 ATP molecule will be synthesized by phosphorylating the ADP and inorganic phosphate which is present in the mitochondrial matrix. Do remember here the major points. First is about mobile electron carriers. What are the mobile electron carriers that are present? Here we do have 2 mobile electron carriers. One is ubiquinone, which is present in between complex 1 to complex 3 and complex 2 to complex 3. And then we do have cytochrome C complex. Cytochrome C complex is towards the intermembrane space, which is from complex 3 to complex 4. Find right about mobile electron carriers, followed by the ultimate electron acceptor of ETS is oxygen molecule, which is present in the mitochondrial matrix, and complex 5 will have F0 F1 channels. F0 will be embedded inside the inner mitochondrial membrane and F1 will be towards the matrix, which will be pumped protons. It will pump the protons from intermembrane space to mitochondrial matrix. Okay. By the end of 4th complex, you have to know the proton gradient which is generated, deficient of protons in this side, matrix side, and high amount of proton concentration in intermembrane space. Thereby, the protons are pumped from intermembrane space to mitochondrial matrix. At the end, ATP is being synthesized. Clear, right? And here one more major point. As such, for 1 NADH molecule, 2.5 accurate ATP is being synthesized. For 1 NADH molecule, accurately 2.5 ATP is synthesized, and for 1 FADH2 molecule, 1.5 ATP is synthesized. Clear till here? Any doubts anywhere, ma? Fine, right? And now, semi-autonomous organelle or mitochondria, which is semi-autonomous in nature. What does that mean, semi-autonomous nature? It can synthesize its own double-stranded circular DNA. It possesses its own DNA and the DNA is double-stranded circular DNA. Fine, right? And it has what type of ribosomes, ma? 70S ribosome, same like that of a prokaryotic cell, whose larger subunit is 50S and smaller subunit is 30S, which is present inside the matrix of mitochondria. Fine, right? They can self-replicate, that is why they are known as semi-autonomous or self-replicating organelles. They synthesize some of their proteins and hence they are called as what? Semi-autonomous organelles. They are known as semi-autonomous organelles. Next is about endosymbiotic theory, which is proposed by Lynn Margulis. Okay, endosymbiotic theory, which is very important. Okay, what is endosymbiotic theory? This clearly states that both chloroplast and mitochondria, have entered into eukaryotic cells by endosymbiosis, has entered into eukaryotic cell by endosymbiosis. What does this mean? Both chloroplast and mitochondria, if you see, okay, they have their own DNA, which is double-stranded circular DNA. They have 70S ribosomes and they can replicate on their own by binary fission. These are the traits which are in similar to prokaryotic cell. These traits are in similar to prokaryotic cell. So, when they are having the same traits of prokaryotic cell. Here, once upon a time, these are the ancestors, they are the independent cells, but in due course of evolution, what happened? Eukaryotic cells have engulfed this mitochondria and chloroplast, and they have become, chloroplast and mitochondria have become part and parcel of eukaryotic cell through endosymbiosis. Fine, right? This is what the theory explains, and this is proposed by Lynn Margulis. What are the evidences that support the endosymbiotic theory? Circular DNA, 70S ribosomes, double membrane and binary fission. Clear, right? These are the major points to be addressed and noted. If you want, take a screenshot now. And then mitochondrial DNA replication, which is also known as D-loop replication in mitochondria. So, how does this happen? Mitochondrial DNA is double-stranded circular DNA is what we have learned, right? Double-stranded circular DNA. So, it has two strands. Yeah, it has two strands. One is heavy strand and the other is known as light strand. One is heavy strand and the other is light strand. Both of them have independent origin of replication sites. The origin of replication site of heavy strand is known as OH region, and origin of replication of light strand is known as OL region. Okay, these are the origin of replication strand sites. So, what happens is, first the heavy strand at the ORI site, it starts replicating a new strand like this. Okay, fine, right? By the time it reaches to the ORI site of the light strand, what happens is, this forms a displacement loop in between OH strand and light strand. The newly formed strand will form a displacement loop in between OH strand and light strand. From the OH region in anti-parallel side of how the heavy strand is synthesizing, in anti-parallel side in the opposite direction, the light strand will start synthesizing its own DNA. As such, both the strands will act as parent strands and both of them will have individual daughter strands. Here, look into the theory once, you will get more clarity. D loop is known as what? Displacement loop. It is a special triple strand region present in mitochondrial DNA. Here, can you see when OH strand is starting synthesizing the new strand over here, here what happens? It forms a triple strand. Fine, right? Locations, if you see, they are present in a circular mitochondrial DNA. It contains origin of replication of heavy strand, which is OH region, origin of replication site, and it contains promoter regions, and it also has regulatory sequences. Why it is called as D loop? When replication starts, a newly synthesized strand displaces the parent strand like this, forming a loop, hence it is known as displacement loop. Fine, right? Now, we look into the steps of replication. So, here heavy strand is rich in guanine, nitrogen base, which is very important to be noted. Heavy strand will have what? Nitrogen base in more content, that is guanine. Light strand, that is L strand, is cytosine rich. Fine, right? Heavy strand is guanine rich and light strand is cytosine rich. Next, each strand has separate ORI regions, I just told you, where OH origin of H strand replication in D loop and OL region origin of L strand replication outside the D loop. Replication model is also known as what? Asynchronous strand displacement model. Look into the steps clearly from initiation, where mitochondrial replication is asymmetric and it is sequential. Here step 1, if you see, initiation at D loop, okay, which has OH region, okay, from the origin of replication region. Replication begins at origin of heavy strand at OH region, which is located inside the D loop. DNA polymerase gamma, this is the polymerase which starts synthesizing the new heavy strand. Okay, this starts synthesizing new heavy strand. New strand will push away from the old strand, forming a displacement loop. This forms a triple stranded region. This is about initiation at D loop. Now you see, in a unidirectional manner the new strand will replicate first. The displaced parental H strand remains single strandedly for temporary time. Okay, for just temporarily the displaced parental strand will remain as single strand. This is the key difference from nuclear replication to mitochondrial DNA replication. Here temporarily it stays as what? Single strand. Now exposure of light strand origin. When replication reaches 2/3 of the genome, when heavy strand replication reaches 2/3 of the genome, origin of light strand becomes exposed, which is located outside the D loop. Okay. And here initiation of L strand synthesis. Now replication starts on the L strand, occurs in the opposite direction from OH strand synthesis. RNA primer forms a hair loop like structure inside the L loop, and DNA polymerase will synthesize the complementary strand here. Completion. Both strands will complete the replication. Two circular mitochondrial DNA molecules are produced here. And here you see both the enzyme and function of the enzyme. DNA polymerase gamma functions mitochondrial DNA replication. DNA polymerase gamma function mitochondrial DNA replication. Mitochondrial helicase twinkle. Okay, this shows what? Unwinding. Next, mitochondrial SSB protein. SSB protein stabilizes single strands. Okay, RNA polymerase primer formation. DNA ligase will join two fragments of DNA. Okay, DNA ligase will join two fragments of DNA. These are the enzymes which are involved in mitochondrial DNA replication. Very important to be noted. You can take a screenshot of this table and remember what are the enzymes that are involved. Next, chloroplast. First you note down the general characteristic features of the chloroplast, then we will move on to the ultrastructure of the chloroplast. Here you see chloroplasts are green color plastids. They are present in many areas of the plant. Unlike the leaves, they are also present in the young stems, sepals, and in unripe fruits the plastids, green plastids are present. One exception, the roots of some plants will also have chloroplast. Example is Taeniophyllum roots. We are learning from our basics. Taeniophyllum roots will be having the chloroplast present in all the autotrophic plants, and chlorenchyma cells as well. They are present in all autotrophic plants and chlorenchyma cells. Their main function, if you see, is photosynthesis, that is food assimilation, is the major function of the chloroplast. If you look into the shape, size and number, shape of the chloroplast can be spherical, oval or discoid. Size of it is 5 to 10 micrometers in diameter. Number is around 20 to 40 chloroplasts per cell. The number of chloroplasts are 20 to 40 chloroplasts per cell. Mesophyll cells may contain 50 to 100 chloroplasts. Clear, right? Mesophyll cells contain 50 to 100 chloroplasts. And then shape, size and number. Shape of the chloroplast, spherical, oval, size around 5 to 10 micrometers in diameter. Number of chloroplasts will vary. In a plant cell, so 20 to 40, in a mesophyll cell or in some other cells like mesophyll cells, it can be 50 to 100 chloroplasts. Now look into the ultrastructure of chloroplast. Chloroplast is double membrane bound, which has periplastidial space in between. In between outer membrane and inner membrane, it has periplastidial space. Okay, it will be having periplastidal space. Fine, right? In between outer membrane, outer chloroplast membrane, and inside it has periplastidal space. And inside the chloroplast, this is addressed as matrix of chloroplast. The matrix of chloroplast is stroma. Inside the matrix of the chloroplast, there is a third membrane system that is developed, which is thylakoid membrane, which is rich in lipopolysaccharides. Okay, the third membrane system which is developed. Okay, third membrane system which is developed is addressed as thylakoid lamellae or thylakoid membrane. Okay, fine, right? Thylakoid lamellae or thylakoid membrane. This is rich in lipopolysaccharides. Fine, right? And these thylakoids, inside the thylakoid membrane you will have thylakoid lumen. These thylakoids will form stalk like structures. The stalk of thylakoid is known as what? Grana. Okay, stalk of thylakoid is known as grana. One grana is connected to other grana by thylakoid, one grana is connected to other grana by stroma lamellae. Fine, right? Thylakoid lamellae or the thylakoid membrane will be having two photosystems, which is photosystem 1 and photosystem 2. They are synthesized from chloroplast DNA itself, whereas stroma lamellae will be having only one photosystem, that is PS1. Clear, right? So this is about grana, stroma lamellae, thylakoid lamellae. Clear till here? And in stroma what are present? Stroma consists of double-stranded circular DNA. Stroma consists of double-stranded circular DNA, 70S ribosomes, okay, and starch granules, lipid droplets. These are present in stroma of the chloroplast. Clear, right? This is about the ultrastructure of chloroplast. Once look into the theory also. The envelope, it has two unit membranes, outer membrane and inner membrane, wherein it is separated by what? Periplastidal space. Periplastidal space is around 10 nanometers. Stroma or matrix is colorless inner fluid. It contains photosynthetic enzymes, circular DNA, 70S ribosomes, and lipid droplets. Starch granules are also present in the stroma of the chloroplast. Functions: dark reactions like Kelvin cycle occurs inside the stroma of the chloroplast itself. Here I have to highlight few things. What are the functions that occur in the thylakoid membrane and in the stroma? Okay. So here if you see, in the thylakoid membrane, and inside the stroma. Here if you see the thylakoid membrane and stroma, yeah. So the thylakoid membrane will be having two photosystems, right? Photosystem 1 and photosystem 2. The functions that it exhibits is it conducts light reactions, that is absorption of light in the form of photons, photolysis of water, and formation of assimilatory power, that is ATP and NADPH. Fine, right? Now in stroma what happens? In stroma dark reactions occur, which are also supposed to be light independent reactions, wherein it has C3 in stroma of the chloroplast, C3 cycle, C4 cycle and CAM cycle occurs. C3, C4 and CAM cycle. Here you look into inside the stroma or matrix what are present, all this we have discussed. These are the functions that occur both in the thylakoid membrane and stroma. And at the end inside stroma what is synthesized? Glucose is being synthesized. Fine, right? What are the products of light reaction here? ATP, NADPH, and as a result of photolysis of water, oxygen is also produced as a byproduct. Clear, right? And then thylakoid stroma. Yeah, here we need to learn about plastid DNA or chloroplast DNA. Okay, plastid DNA or chloroplast DNA. Plastid DNA or chloroplast DNA is also known as cpDNA or ctDNA, circular double helical DNA. Okay, circular double helical DNA, which is located inside the stroma. The origin of this chloroplast DNA is cyanobacteria. This is via endosymbiotic origin, and about endosymbiotic origin we have already discussed. Each chloroplast will be having how many DNA molecules? 10 to 100 copies of chloroplast DNA are present, which has 120 to 200 kilobase pairs. They are smaller than the nuclear DNA, larger than mitochondrial DNA. If you have to weigh the molecular weight of chloroplast DNA, mitochondrial DNA and nuclear DNA, here will be the ascending order. First is chloroplast DNA, first is mitochondrial DNA and chloroplast DNA. And then nuclear DNA. Fine, right? Mitochondrial DNA, chloroplast DNA and nuclear DNA. Nuclear DNA is of larger content. First is mitochondrial, chloroplast DNA and nuclear DNA. This is the right order. Okay. Chloroplast DNA is smaller than nuclear DNA, but it is larger than mitochondrial DNA. Clear, right? This is the ascending order. Remember, very important. Fine, right? Next, genes which are present like chloroplast DNA codes for photosynthetic proteins, where Rubisco is the largest, Rubisco larger subunit is synthesized. Photosystem 1 proteins, photosystem 2 proteins are synthesized by chloroplast DNA only. Cytochromes, ATP synthase, that is F0, F1 channel is synthesized by chloroplast DNA itself. And then RNA components like mRNA, rRNA, tRNA. From rRNA again 16S subunits, 23S subunits. These are all synthesized by the genes that are present in the chloroplast DNA. Very important to be noted, remembered and focused. What are the proteins and components that are synthesized by chloroplast DNA are very important. And then functional significance and comparative analysis between mitochondrial DNA and chloroplast DNA. Functional significance here, if you see, Rubisco enzyme, it will partly synthesize, Rubisco enzyme is partly synthesized by chloroplast genes. Remaining subunits are synthesized by nuclear genes. Mitochondrial DNA and chloroplast DNA, mitochondrial DNA is purely maternal inheritance. It is inherited from the female parent, from the female cell, that is from egg cell, right? Whereas chloroplast DNA will follow different patterns of inheritance. It can be maternal inheritance either from the mother, or it can be the paternal inheritance from father, or the inheritance can be biparental, sometimes from both egg cell and the sperm cell. Next, transmission is from mother, and here transmission depends on different species. Next, this is common in animals and in some plants. Here chloroplast DNA, mostly plants or algae. Variation, Heteroplasmy is common in mitochondrial DNA inheritance. Variegation is common in chloroplast DNA inheritance, that is giving out non-green parts of the leaf. Next, famous example here is human mitochondrial diseases. Here, mirabilis leaf color which shows variegated leaf expression. Pedigree is from mother to all children. Here this depends on the type of plant cross. Any type of inheritance is possible. Genetic ratios are non-Mendelian both for mitochondrial DNA and chloroplast DNA. This is the complete information about mitochondrial and chloroplast DNA. In our next class, we will be looking into plasmid DNA, followed by we will focus on nuclear structure, nuclear DNA, chromosomes, Polyteny-Lambrusche chromosomes. Okay, Polyteny and Lambrusche chromosomes. We will fix this out in the next class. I hope till this session, that is mitochondrial DNA and chloroplast DNA, is clear to you. If you are having any doubt, you can ask me. Fine, right? Yeah.